third section of our day today.
Recording
that would be
in
great.
Progress.
So as you're making your way back to your seats, big welcome back. Thank you. Thank you all for being here. This has been a forum that's about a year plus in the making, and we couldn't be here without all of you to support us, support looking at these environmental topics, and certainly to our speakers, our amazing panel of experts that I have to say it's kind of impressive to realize all of these experts have been in plain sight all this time, and yet we're finally able to bring them together to talk about this really important issue today. My name is Sara Walling, and I am the Water and Agriculture Program Director at Clean Wisconsin. Clean Wisconsin is a statewide environmental organization, some 20,000 plus members and supporters strong, who works on all areas of clean water, clean air, and clean energy. So, as you can imagine, this issue of neonicotinoids and its impacts on our environment, our water systems, our soil systems, and us as human systems sets pretty squarely within my wheelhouse at Clean Wisconsin. In addition to all the thank yous for you guys being here, thank you to everybody who's online. We have at least as many people signed up online as are in this room, so that's fabulous to see the level of interest in this issue. Also, a thank you to UW and the Nelson Institute and all of our other supporters for helping us put on this event, especially the Wisconsin DNR and their Surface Water Grant Program for making this event possible today. A couple of reminders I guess I want to make you all aware of is certainly remember that all of these talks, including the slides, are being recorded, and so will be available on the Clean Wisconsin YouTube channel within the next few days after the closing of the forum. So everybody will have access to all the slides. So don't feel like you always have to whip out your camera and take a picture where you want to make sure you retain that info. Also has been scrolling through the reel on and off throughout the day, and will again is a QR code for our survey. We'd highly appreciate if you could pull out your phones when that does come back up and complete the survey toward the end of the day to let us know how we did, let us know what issues and what components of the sessions today really raised flags or interest for you, and then what your interests are in learning more about this and what more we can do as all of our organizations to continue to amplify awareness about some of these issues in our environment. Last thing I'd like to say is, all good things, including this conference, often happen over a beer at the Union, and so, like I said, this forum is no exception. Andy Morton came up to me at the Memorial Union a year ago last summer with the idea of putting on this forum, and we hit the ground running from there and brought in a lot of other supporters, most of who are in the room today. And so we'd like you to join us after the sessions here today at 4:30 across the street in the Union South this time at the Set. So if anyone is interested in a little social hour afterward and meeting up with any of the speakers who are able to stay behind, and then any of you attendees, we'd be happy to sit and have a chat, talk a little bit more about any of these, answer some of those burning questions that you didn't get answered during the sessions across the street at 4:30. So we hope you can join us. And with that, I have the prestigious honors of welcoming our next set of panelists. And so I am at the age where these have to come on for this part, but I am welcoming the three speakers to continue our discussions on the impacts of neonics to biota. First, we will have Dr. James Crall joining us from the Department of Entomology here at UW-Madison. His presentation is on understanding the impacts of neonic exposure on bees. James received his bachelor's degree from Swarthmore College and his PhD from Harvard University in the Department of Organismic and Evolutionary Biology. He's interested in behavior and ecology of insects and how they're impacted by rapid environmental change. His research group at UW-Madison studies bee health and plant-pollinator interactions with an applied interest in supporting pollinators and pollination. His lab develops low-cost, scalable technologies for experimental automation, data collection, and analysis, including AI and computer visioning. He's especially interested, though, in how these emerging technologies can help us better understand the impacts of environmental change and pesticide exposure, in particular on bees and other pollinators. After Dr. Crall, we'll welcome Dr. Gavin Dehnert. He is with the Wisconsin Sea Grant, also here at UW-Madison, an emerging contaminant scientist, exploring neonicotinoids in aquatic ecosystems with a deeper look on those impacts on freshwater fish populations. Gavin received his undergrad degree from the University of Miami in Florida, and a BS in marine science and a BS in biology. He followed that up with a PhD from UW-Madison in integrated biology and aquatic toxicology. Also doing some postdoctoral fellowship with our Wisconsin Department of Health Services, where he helped set recommended health groundwater standards for compounds, including some PFAS work. Following that, Gavin became an emerging contaminant scientist with Sea Grant, where he conducts research on the impacts of emerging contaminants on aquatic ecosystems largely. And then last but certainly not least, we have Dan Raichel from the Natural Resources Defense Council, where he is the Director of Pollinators and Pesticides and Nature Programs for NRDC. He's going to be talking to us about the harms to birds and human health. Dan focuses on protecting our nation's bee populations from the ever-growing threats to their health and existence, in particular the use of bee-toxic pesticides. Before joining the Nature Program, he co-directed the NRDC's Community Fracking Defense Project and was an advocate for the cleanup of industrial pollution in the New York region. Dan has a bachelor's degree in English from Cornell and a JD from Columbia Law School, and he works out of the Chicago office. And while he didn't perhaps travel the farthest today, he probably had the most challenging commute in that his bus was canceled this morning. So he had to get into a car and made it here just in the nick of time for this session. So thanks for the extra stress, I guess, that went into his system today too. So with that, I would like to welcome Dr. James Crall to the stand to give his talk.
Great, thanks for that wonderful intro, Sarah. And huge thanks to all the organizers. And I just want to add one, I think, very important piece of biographical information to know about me, which is that I happen to have bought a house that is 2 doors away from Mike Miller. So it's been fun to have conversations informally and the sort of important, very Madison experience, I feel like, having good street-side, spontaneous conversations about biodiversity and neonics. So yeah, really fantastic to be here. And what do I press down? Is that the... Great. Okay, yeah, so I'm an assistant professor in the Department of Entomology here at UW-Madison. My lab is just down the street, and we really focus on thinking about pollinators, bees in particular, and I'm going to take you on a little bit of a journey into the world of bumblebees in particular as kind of a model system for thinking about pollinator health. And we are interested in both in the basic biology and ecology of those organisms, but also thinking about how that really fundamental understanding can help us think about bigger picture questions in biodiversity and ecosystem functioning. So I'm going to be talking more about the terrestrial side a little bit, but I really want to start by reminding us of the astounding diversity of insects in particular that Mike highlighted this morning. One of the amazing things is we actually really don't know how many insects are on the planet. The estimates range from like 1.5 to 10 or more million species, and that's because only about 1 million of those are actually described, but they're an astounding amount and really staggering diversity of biodiversity sets just in insects. And of course, we're familiar with some of the negative things that insects do, like biting us or eating our food. And of course, this is in many ways why we're here, thinking about insecticides and their impacts in particular. But insects also do an astounding amount of really important things that we care about a lot. That includes providing food for fish and other things that we consume. Themselves, they are often predators and provide sort of predation control services agricultural ecosystems. And then one thing that we care about a lot that I'm going to talk about in particular is this bottom row shows this amazing ecological interaction. And that is the coevolution between insect pollinators in particular and the flowering plants that depend on them. And this amazing interaction is really important for us, just as a reminder of things you probably know already, but the vast majority of flowering plants on the planet depend to some extent on animal pollinators. And that's not exclusively, but largely bees and other insects are really critical to those pollination services. And just from a selfish human perspective, we care about that plant-pollinator interaction a lot from a crop pollination services perspective. So a lot of fruits, nuts, and vegetables, including ones that we grow here in Wisconsin, are highly dependent on insect pollinators. A lot of existing yield gaps are actually likely due to insufficient pollination. So within the US and also globally, lack of enough pollinators is actually a really agronomically important factor, and potentially increasingly so. And we know that although a lot of pollination comes from honeybees, diversity we recognize is incredibly important. So we have a lot of native pollinators. Honeybees are not native to North America, but they're very important for agricultural productivity. But those other 4,000 species of native bees we have in North America, really important for the delivery and stability of crop pollination services. So just from a selfish human perspective, we care a lot about crop pollinators. And so given that, I think this is at least the second time you'll have seen this slide, or at least the graph on the left today. There's been a lot of concern over increasing recognition that we might be seeing really dramatic declines in insect populations. So these are some data that in some way kind of kicked that off from about 7 years ago. Long-term study in Germany looking at overall biomass declines and diversity declines in insect populations. So really kind of started ringing the alarm bells about overall population declines, and there's been a lot of work in the years following those initial findings trying to unpack the drivers of those. What are the factors that are actually driving declining insect populations? And what I just want to highlight on the right there, you probably can't see that, but agriculture, one of the really absolutely clear and key stories that has emerged from that is that agriculture is a very important driver of insect population declines, and we know this really well. There's great data on this. Now, the specific agriculture means a lot of things, right? It means simplified landscapes that have different food resources, physically distinct landscapes, but of course it also means a lot of what we've been talking about today, which is increased agrochemical inputs, including insecticides in particular. So I'm going to focus, surprise, surprise, on some work that we've done focusing on neonicotinoid pesticides in particular. And from the biologist perspective, I just want to remind us what these things do, right? So they are agonists of nicotinic acetylcholine receptors, which you can think of that as in some ways being the backbone of the insect's central nervous system. They're incredibly important. It's the primary excitatory neurotransmitter in the insect's central nervous system. So kind of just really important for everything is the takeaway in short order. And again, a graph that you've seen a couple times already, but these have increased a lot in their use in agriculture within the US and also globally. And of course, they are persistent and widespread in the environment. So, you know, one sentence description, but you've now seen a half day of talks describing the extent and, real effect of those, and really fantastic work actually documenting the complexity of that contamination. So I want to kind of think from the bee perspective a little bit and think about why that contamination matters for terrestrial insects and pollinators in particular. And one of the reasons it really matters, if we've talked a lot about, for example, seed coating, right, and the systemic protection that plant tissues get and crop tissues get throughout the growing cycle. One of the challenges, even though that's great from a crop protection perspective, that also means that those compounds that are systemically moving through the plant show up in nectar and pollen that bees consume, and then they can take back to their colony, where they're often then feeding and transmitting to other nest mates, and that might disrupt critical behaviors, and things that the bees need to do outside of directly killing and causing lethality in bees. So how widespread is that exposure from the bee perspective, right? So we've heard a lot about overall environmental contamination. These are data thinking about that contamination through the bee perspective a little bit. And this is a delicious sounding study from a few years ago that basically just went and collected as much honey as they could from across the globe. So sort of, in many ways, using honeybees to sample the environment, right? So doing biased sampling for the stuff that bees like, which is nectar and pollen. And the white samples there are ones that don't have any detectable neonicotinoid. You'll notice those are in the vast minority. So about 3/4 of the honey samples from across the globe have detectable amounts of neonicotinoids, often multiple neonicotinoids, and often at concentrations that we know have physiological impacts. So exposure is quite widespread for probably the majority of bees on the planet. So what are the impacts of that exposure? So there have been now some really nice studies. This is from a large-scale field study following the ban in the EU, looking at the impacts where they looked at whole fields, either treated with or without, in particular, a seed treatment, and found evidence for negative effects on wild bee populations and growth. So on the left here, it's just showing effects on wild bee, these are solitary bees, overall density, and also nesting rates. So, you know, the most striking error bars you can see, 0, right around 0, absolutely no nesting of some of these bees. In the middle, this is showing impacts on bumblebees. And so these are colonies that were put out and they tracked growth over time. And basically what you see is those colonies didn't die immediately, but growth was impaired. So we see less reproductives and also fewer workers. Interestingly, in these studies, when you put out honeybee colonies in the same fields, exposed to the same conditions, we actually see relatively little effect. And these patterns have been borne out in other studies. And a pattern I'm not going to talk about too much in depth today, but it could well be that honeybees are the least sensitive. Out of the 20,000 bees on the planet, honeybees might be the most robust. They're our main model for thinking about sensitivity of bees, which is its own problem. But especially for native and wild bees, evidence that exposure at the field level does drive decreased populations and impairs colony growth. This is from, again, experimental fields, but what about if we use some of these great emerging long-term data sets to examine similar patterns at the sort of geographic scale? So this is great data that just came out looking at the declines of what was once a very common bumblebee species, the western bumblebee, Bombus occidentalis. And then, so you can see their range has declined really dramatically. And then by using historical exposure data, what this study found is basically the declines in that population are driven partially by climate, but also importantly by particularly the nitro-containing group of, so the particular toxic neonicotinoids. Okay, so I think in sum, what we know pretty well now is that there's lots of neonic exposure. That neonic exposure gets to bees, and that exposure can drive declines in populations and declines in colony growth. But these are also mostly at concentrations that are not toxic. So what I want to sort of think about a little bit with you today is, well, what are actually driving those declines? What are the mechanisms? And if we better understand some of those mechanisms, can that help us build a system that can better predict, for example, risk of new compounds that we put out in the environment, and also understand the complexity of these responses? And a couple things that I want to really highlight, thinking kind of from the perspective of, let's say, a bee colony that has to do a lot of stuff, right? And one of the challenges in understanding some of the compounds that are really emerging in this field is we focus a lot on lethality. But again, these are neurotoxins that have a lot of impacts on the nervous system well below, like, as you know, you don't need a toxic dose of alcohol to impair your capacity to function in important ways, right? There's lots of stuff you don't want to do or you can't do when you're consuming low subtoxic amounts of neuroactive compounds, right? So these sublethal effects have been harder to characterize, but we know really important. And also we know that there's not just one stressor at a time, right? So not only we've seen lots of data on this today, but not only are bees and all the other insects exposed to neonicotinoids, that's combined with other neonics and other things like fungicides, herbicides, so lots of different agrochemical exposure, and also combined with stress from the environment, like nutritional availability or temperature. So, you know, these organisms are coping with a complex suite of stress out in the environment. And these are some of the things that really we've come to recognize are critically important, but make it really hard to understand, why different groups might respond differently, or we see different impacts in different places. Oh, that's way choppier than I want it to be. Okay, imagine this is a beautiful prairie field with a smooth bumblebee wandering through it. You can use your mind's eye for that because you've seen it. You've seen it a lot. But so I'm going to bring you a little bit into the world of bumblebees to think about as a model system for thinking about those complexities. So again, mind's eye, a bee flying around this environment, sort of a windy hill. This is on a hillside in Vermont. And this is something we're really used to seeing, right? But there's actually a lot of amazing organismal ability wrapped up in that moment, right? That bee is, they're like remembering complex landscapes. They learn really well. They remember where food is. They're flying through a physically complex environment that is also moving. Like imagine trying to fly a helicopter through skyscrapers, but those skyscrapers are also like flopping around, and you have to get food from the top of the skyscrapers, right? It's an amazing challenge that is way beyond what human technology can do. And they're doing all that with a couple orders of magnitude fewer neurons than we have, right? And they're doing it with all the energy they need on their back. So there's all this amazing adaptation associated with being able to actually move through that environment and get food. And what that food is doing, is this one gonna play? We'll see. A little better. Is really fueled at taking that food from flowers in the environment and taking it to, in the case of bumblebees, usually a hole in the ground where we then have no idea what's happening with it for the most part, or we're not used to seeing it because it's this whole world of the social life of a colony where those resources are getting turned into new larvae. They're being moved by other workers and basically being turned into colony growth and eventually evolutionary fitness, right? So what I really want to highlight is there's a lot of stuff that has to work right all together for a bee to be able to turn food into population growth. And any one of those cruxes might be impacted by these compounds and drive long-term impacts on populations. And it turns out we have a lot of evidence for impacts on different parts of that cycle. So if we start sort of outside of the bee foraging, we now know quite well from decades of study that exposure to neonicotinoids can, for example, impact overall foraging. So this is just showing, if you look at the number of bees that are carrying pollen, those are impacted by imidacloprid exposure. The capacity to learn what flowers have food, that is also impaired by neonicotinoid exposure. The ability to navigate through the environment, so if you strap a little tracking device under the back of a bee and watch how they can move, they home less well. And all of that sums up to an impaired ability to bring resources back to the colony, right? So foraging levels, learning, navigation are impacted, and then less colony intake. So that can be an important driver of colony losses or colony declines, something that we've been really interested in the lab, and I already got the 5-minute warning, and I'm not extension, but I still do talk too much. So it's something that we're really interested in the lab is thinking not just about what happens to bees when they're out in the environment and where we see them, but what happens in that other half that I showed you. When you bring resources in and those resources have to get turned into colony growth, how might that be impacted? And so this is the life cycle of a bumblebee colony a little bit. Unlike honeybees, they start solitary. So they start as a single mom starting a colony off. You'll actually see the big queens foraging in spring until workers emerge, and eventually the colony will grow through the course of summer until in the fall around here. So this would have happened about a month-ish ago, a few weeks ago. Reproductives come out, mate. Everyone but newly emerged queens die, so the whole population goes back down to just queens over winter, and then they start the cycle again. So what might be some of the impacts on, we know that those food resources that are contaminated with neonicotinoids are being brought into the colony. What might be impacts on within nest behaviors? That has been more of a black box. And so we developed some fun tools that I'm going to walk through very quickly, where we can basically use some computer vision techniques to track individual bees and use that to quantify behavior to figure out whether or not they're doing things like nursing, how active they are. Turns out there's all kinds of interesting patterns of each bee has her own personality within these colonies. So there's lots of interesting, really fun biology, but you can also do slightly more depressing things like expose them to imidacloprid, but use these tools to look at the impacts on these more subtle behaviors. And so what we can do is look at individuals, expose them to, in this case, acute exposures of imidacloprid, which you've already heard a lot about, and then we can look at changes in behavior. And if we look at, for example, overall time spent nursing, we see, at least at our 1 nanogram exposures, which is kind of on the upper end of environmentally realistic exposures, so what we expect a forager to consume in a whole day at environmentally realistic concentrations. We see reductions in nursing, reductions in overall activity, shifts away from the nest center, so bees are kind of more on the periphery. And also we can map out the social networks of colonies, sort of who's interacting with who, and they become less social. Basically, they get pushed to the periphery and interact less with nestmates. And so, again, impacts on, these are not lethal effects, but important impacts on behavior. Now I'm going to start sprinting. We can do the same thing with a robot and track for a long time. And basically what we find are similar reductions. So you see reductions in nursing, activity, and overall social interactions. Those impacts actually have a circadian rhythm. Interestingly, they show up overnight and not during the day. So one way in which these effects might be a little bit hidden. And they can also disrupt, if we look out in the field and put temperature probes in colonies, what we find is that control colonies are really good at actually running a temperature, and that capacity is impaired under imidacloprid exposure. They can't do things like restructure the nest. They can build a wax canopy. That capacity is impaired. Basically, like putting a blanket on yourself, that capacity seems impaired under imidacloprid exposure. So also these sort of long-term chronic exposures have important negative impacts. I'm not going to look at the timing. I know I'm over. Can I have one more slide? Okay. And I think this is just an important point, so I'll go through this very quickly. But another thing that we found is as we watch those colonies develop, basically what we found is if you look at the same suite of exposures in young colonies versus older colonies, younger colonies are more sensitive. So around here, that would be early in the season, right? So usually we work in these mature colonies, but those early season colonies, it turns out, are more sensitive, which probably shouldn't surprise us. So overall, I think we have now compelling evidence that there are very real important impacts on pollinators, even that aren't lethal. And I think the ways in which we study the impacts of neonics, nice room temperatures, well-fed bees, middle of the day, mature colonies might be systematically biasing us against picking up what the impacts under real complex conditions are. And I think something that we're really excited about, and this is kind of where I'll finish, is I think we need better technology and monitoring tools to be able to actually understand these real-world impacts. And also mesocosm style, be able to bring more complexity to the lab and also go out in the field. So in my lab, we're really interested in taking these tools and making them cheap and easily available, and moving out into the field where we can understand what colonies are doing in real-world environments, and also thinking about whether or not maybe we can use emerging AIs and these sort of low-cost, increasingly available technologies to monitor things like endangered species. And with that, last thought is that we know that exposures are, sorry, I know, right? Yes, I know. But really want to highlight that one of the reasons we need that is it's not just neonicotinoids, right? The neonicotinoids are an important part of the complex suite of stress that bees are exposed to. Okay, with that, I'll stop talking. Thank you.
Great, thank you. Now I will welcome Gavin Dehnert to the stage as he makes his way up. Thank you all again. We'll hold questions until the end, so we should still have, even with everybody probably going a little bit over in this session, I think we'll still have plenty of time for questions at the end. So.
Don't worry, mine's less than 20 minutes. We'll be on time. Oh, no. Yep.
All right, well, we're waiting for that to get uploaded. I guess just to, again, do a couple of thank yous and shout outs to some of our group here today and also those who couldn't be. But I have to say I'm extremely thrilled, obviously, that this is being, this is actually happening today. This is, like I said earlier, about 1.5 years in the making. And so a lot of work has gone into it. But in addition, it's fabulous to see the diversity among this audience. There are everyone from fly fishermen and women to students to very in the weeds researchers to applied scientists and agronomists to our policymakers in all of the state agencies that have a role to play in this issue area. So couldn't be happier for everybody to be here. And thank you especially to Carla Romano, who is our tech star over here running all of the slide decks on the side for us. So I think we're ready for you, Gavin.
Perfect. All right, well, was under 20 minutes. Now we might be back up. Forgive me, I might have to jump back and forth through a few slides, but we'll get through this. So again, my name's Gavin Dehnert. I am an emerging contaminant scientist with Wisconsin Sea Grant. And the first disclaimer I want to tell you guys is I study PFAS, microplastics, and pesticides. And I don't really study neonics, but my main focus in this world is kind of understanding how different contaminants impact aquatic ecosystems, and particularly fish. So for today, we're going to kind of explore neonics in aquatic ecosystems, but really focusing a little bit deeper on freshwater fish, and then I even threw in a little bit of amphibian work just to spice it up. So, there we go. So we're going to jump down here. So, there we go. Okay, so I'm not going to spend a lot of time because we all know about neonics here, but I just want to give like a spoiler alert. All the same neonics that we're learning about and how they have impacts on, aquatic or terrestrial insects and the bees that we just learned about, they're the same kind of big ones that have been studied for fish species. And why do we really care about them is we use all of these neonics, right? They're used on agriculture again, which we've talked a lot about. They're used in homes, lawns. They're used kind of all over the place. And because they're water soluble, they do make their way into aquatic environments, and specifically into surface water or lakes across the state of Wisconsin. And that's really important when it comes to fish because fish can't leave a lake, right? They don't have the ability to fly away. They can't kind of get out of a system. Once the contaminant is in the lake, they are going to be fully exposed for as long as that contaminant is there. And so again, as we've kind of talked about, you're right, these insecticides, they were brought in for a reason, right? They have target species that they're really focusing on, whether that be flies, ants, etc. But again, as these, get into the aquatic ecosystems, they can have these impacts on non-target organisms, which is a lot of like the stuff that we in our lab study. And mostly, we're going to focus on the three of them here that are different fish species, mostly the freshwaters, and then a little bit of the amphibians. So we're going to take a big step back, because when we start to think about, all these impacts on terrestrial insects, there's a lot of research that's done out there. But when we, kind of look into it as like, what are those, known published literature studies that are on, impacts on fish, we have a much lower amount of literature that exists out there. So just to point out here, this on the y-axis, these are the number of articles that have been published. X-axis, these are our different types of neonics that we've kind of exploring. And so as you can see right off the bat, there's a little less than 150 different studies that have even been published on fish, which we can talk about a little bit of maybe the why. But the one I want to kind of focus you on is the main driver of all of this stuff, right, is imidacloprid. So there's a lot of other different, neonics where there's little to no kind of research that's done on them. So while today I'm, going to be talking about what do we know about the impacts on fish, what I'm really going to try to implant in the back of your head is that there's a lot of missing data, and there's a lot of things that we don't know about this to start. And again, I just wanted to highlight this for imidacloprid. So, imidacloprid was first introduced, in 1991, got registered by the EPA in 94. But you notice the first studies when we start learning about fish, they don't happen until 1998. So you can see there's this, gap to kind of start. But the big one to point out is majority of the research that's been done on, the impacts on fish has really been done in the last 4 to 5 years. And a lot of this still is going to be on these like big picture, like, lethal and, some sublethal impacts that happen. So as I said, we're going to take a big step back, right? When we're thinking about, what do we actually know about the impacts of these neonics on fish species? And believe it or not, a lot of the literature focuses on this thing called the LC50, or the lethal concentration to kill 50% of a population. And so a lot of this work, right, is going to be focusing on that to start. And then eventually, towards the end, I'll dive into a little bit more of what we know about these sublethal impacts, which I'll give you a little spoiler alert, it's not as much as we know about the lethality stuff. So, for starters, the first kind of really useful information to kind of come out goes back to about 2005. And when we're thinking about these non-target organisms that aren't insects, the three that we're going to kind of focus on first, right, is we have our mammals, birds, and then fish on the far right. And the reason I kind of point out this graph as a good starting spot is so these are the lethal concentration to kill 50% in the fish here of two different species. These are trout and carp. Now, I don't expect you to be able to read all of this stuff, but the big thing I want to also point out is the concentrations that we're looking at here are in parts per million. So the lethal concentration for some of these, is well above 100 parts per million, which, as we know, is a lot higher than, what we're seeing in our aquatic environments right now. Kind of diving into this, and as I had mentioned, a lot of this research has been really coming out in the last 3 to 4 years, 4 to 5 years. And, again, the point isn't to kind of read through this graph, but some big highlighting things to show is they've started to look at some other different species with these acute studies. And these acute studies, again, right, these are 96 hours. They're exposing these fish for, 4 days in an environment, seeing how much of them, or seeing what the lethal concentration can be. And again, this column right here is really pointing out that, two big things, right? We're in parts per million again. We are, the lowest one that we're seeing here is somewhere about 30 parts per million, so much higher. But we are also starting to see a little bit of a range that's happening. And the reason I point this out is so, unlike the bees and the insect world, we do also in fish have about 27,000 different types of fish species, so we do have, some variety. And what we know is that some species are going to be more or less sensitive to some of these different insecticides. And in particular, again, though, the focus is like we're at much higher concentrations than we are seeing in the environment. The other thing I would just want to kind of highlight here too is that the rainbow trout, when you're seeing some of these things, this is a list of like all the different types of neonics. You're not only seeing different sensitivity based off of the different species, but you're also seeing different sensitivities to each of these insecticides, which again becomes important when we're thinking about, there was, over 100 studies that are done on imidacloprid, but some of the other ones had much, much lower. So we're still missing a lot of the gaps that exist out there. And then a couple other big things to highlight is there's been a couple different studies that have explored different life stages. So as we start to think about embryo stage to the larval stage, juvenile, and adulthood, each of these life stages can have different sensitivities. Importantly, this kind of top graph or top portion of this table really kind of focuses on this idea that there isn't a clear highlight to say maybe the embryos are the most sensitive, maybe the juveniles, but actually kind of points that maybe the most sensitive is some of these adult life stages, which is a lot different than some of the other pesticides or herbicides that have been used and thought of before. And as promised, I'd throw in a little bit of the amphibian work here. And a couple things to kind of point out about the amphibians is when we thought there wasn't that much about fish, there's even less that's done on amphibian work that's out there. But similarly to the fish work, our concentrations that we're seeing that kill, that lethal dose at 50% are pretty similar to our fish. And again, you're kind of in that, 20 to 100 parts per million range, so much higher. So again, it's kind of hinting at this idea that at least for acute lethality, we're probably not going to see too many impacts on our amphibians or our fish species. But again, like I said, part of what my goal up here today is kind of not only tell you what we do know, but it's really kind of highlighting all that we don't know. And the biggest thing I want to first kind of highlight is the missing of the species, right? We're in Wisconsin. We all know about our walleyes, our northerns, et cetera. And majority of our fish work that's done kind of focus on these rainbow trout, carp, and then our two model organisms that we use quite a bit, which are going to be those zebrafish and fathead minnows. I did kind of come across one singular study that looks at a little bit of yellow perch, but very minimum. And then second, to highlight this very limited amount of studies on amphibians. As I said, we're in Wisconsin here. These are some of our main species that, we like to kind of think about. Everybody here loves the walleye. Everybody has had a walleye fish fry. There hasn't been any kind of studies that look at neonics on this, which is kind of can be slightly concerning when we have thought about the walleye can tend to be one of the most sensitive species when it comes to fish, especially for some of the other herbicides and pesticides that are used in our waters around here. The second kind of missing data that really exists out there is this idea of active ingredient versus commercial formulation. So for those of you who don't know, when we're using these formulations out in the environment, these commercial formulations have inert ingredients, right? And these inert ingredients can be surfactants, they can be other things that can kind of help it cross membranes, last longer in the environment, and et cetera. And sometimes they can play a role in toxicity levels. So a lot of the studies that do exist out there, either focus mainly on just a single active ingredient, and then there is a few that do explore the commercial side formulation. And then there's one study that's really looking at potentially, is the commercial more or less sensitive than the active ingredient? But again, this is a huge gap in the literature that's out there. And then if I wanted to give the biggest takeaway when it comes to the lethality behind this is to more focus in on, again, of the levels. So when I'm talking about the impacts of, all of these being, 100 parts per million, we're really talking about in the nanograms per liter and the sometimes even into the the micrograms per liter, but these are, 100 to 1,000 times fold lower concentrations, so most likely not going to be having large impacts on survivorship right away. So some of the big impacts of, again, with what we do know is the fish and amphibians do kind of show some of that similar toxic levels at this chronic. There's no really life stage that is more or less sensitive that's coming out at the beginning. And then most importantly is that not all of these neonicotinoids are going to have the same concentrations, have the same impacts at the same levels. But we know that neonics last in the environment for more than 96 hours, right? They have longer, skip ahead. They have longer half-lives. They can have very long half-lives in soil. And then, they can have half-lives in aquatic ecosystems sometimes up to 75 plus days. So once you start thinking about this, we should also not think about just acute exposure, but also like a chronic exposure to these things. And so there is some literature that kind of explores this. And mostly what I really want to point out about these things is you're going to see a lot of the same kind of concepts come through. But the first one, and maybe the most important, is again, when we start exposing them to chronic exposure, our levels of when we see, LC50s or lethality is going to drop to, less than 1 part per million. So now when we chronically expose something, we're exposing them, lower doses will have lethality impacts, which makes total sense. So it's not surprising to see that, but again, that 1 part per million is much lower than, or much higher than some of those concentrations we're seeing in the environment. And just to kind of add into it, because we have amphibians as well, again, exact same things that we're seeing where, those chronic exposures, you start to see impacts at lower levels, but you're really not seeing these at levels that should cause concern for, complete population impacts. Kind of one important kind of aspect about this is, so some of these species that are out in the environment, like the fathead minnow is a prime example, is there can be transgenerational impacts to these neonics in the environment. So some of our species, when we're thinking about fish, right, they spawn once a year, but we have some species like the fathead minnow that can become sexually reproductive within 3 months. And so what can happen with that is when there's multiple exposures over multiple years, potentially we could start to see some, transgenerational impacts. But again, none of these studies have actually really been done in the neonic world. So again, another large gap. But with some of these chronic studies that were kind of done, it points out a little bit of some of these potential sublethal impacts. And again, as we had just learned kind of the impacts on terrestrial, there's some potential behavioral impacts that we're starting to see. So of the sublethal impacts that have kind of been explored, have really focused a lot on locomotion and prey capture and potentially predator avoidance. So these are sublethal behaviors that can be kind of mapped similar to what we just learned about with the bees, but with our little larval fatheads as well. So I'm just going to kind of point out a couple two different papers that kind of start to show this. And again, the first one is done by a collaborator down in La Crosse, Tisha King-Hayden, where she's starting to look at exploring how, what are some of these sublethal impacts. The graphs here on your left are really going to focus on once they hatch out. So these are larval fish that are exposed to different concentrations of thiamaloxifin. And on the y-axis, we have different behavioral things. So on the top, we have latency. So once they are stimulated to a vibration, it can track how long it takes for that fish to move. How fast are they moving after? And then something along the lines of like the total escape. And as you can see, the top graph is kind of showing that the higher the concentrations get, we're seeing a longer time until they move away. And similarly, also a decrease in how fast they're able to move away. So again, these are two potential behaviors that could have impacts on their ability to escape a predator. Another one I just want to highlight here is looking this time at imidacloprid on fathead minnows. Again, they kind of focus on the same three big things where they're looking at sublethal effects on behavior. Again, we have that latency response on the top right, our burst speed, and our total escape response. And they're following kind of very similar aspects, right? Is regardless of if it was imidacloprid here, the latency response increases with higher concentrations, and our burst speed is decreasing with higher concentrations as well. And one final one with behavior here is they've looked at this with adult zebrafish, and I thought this was a really interesting study to highlight because they're looking at not only movement, but the memory to be able to know where the food is being added into the tank. So what they would do is they would always add food to a certain spot of the tank, and over exposure over time, kind of measure not only what their speeds are to moving to it, what their average speeds are, average distance to get to it, the maximum distance, etc. They were also able to kind of find, similarly, that they weren't able to remember where the food would always be coming in. So it's kind of an interesting finding. Let's see what else. Oh, and then the very last thing, perfect, and I promise, I'm actually going to skip to the fun one, that I just want to kind of highlight about this is, so again, we're talking about single exposure to a single neonic for all of these studies that are out there. As we know, there's no such thing as one neonic out in the environment. There's not only just neonics out in the environment, there's PFAS, there's pesticides, there's everything else. So a lot of the studies are really only focusing on one, and we know that mixtures can play a huge role in this. And just to kind of highlight this is some of these can actually have an additive effect. So if we were combining two different neonics together, we can see an increase in the response that is being impacted. But sometimes if you're combining them together, we're actually going to see an antagonistic impact. So not only is it important to study these individually, but also how do these mixtures play a role in everything. And with that, I will just leave you with my favorite photo of Norman the Northern, who has two heads. So yeah, thank you. Perfect.
And he still managed to keep it at time, so fabulous job. Lastly, but I would like to welcome Dan up to the stage to talk to us about birds and humans. We'll take it over.
Hi, everyone. Guess I'll wait for my presentation here, but I'm glad I could make it. The bus debacle this morning was, here we go. Yeah, my name is Dan Raichel. I work for the Natural Resources Defense Council. I am an attorney, and I direct our pollinators and pesticides work. I should note, too, NRDC is a national environmental nonprofit. I'm going to talk a little bit more broadly today than advertised. I'm going to talk about neonic impacts to biodiversity at large and also some of the threats to human health. So let me get started and explain why we're so focused on this particular class of insecticides. It all started, oh wait, I got to point it this way. It all started in the mid-2000s when beekeepers in Wisconsin across the country suddenly started to see their annual losses of colonies shoot through the roof. Up until that point, beekeepers were typically experiencing 10 to 15% losses every year, mostly due to winter cold. And those numbers jumped to 30, 40, 50% virtually overnight, and also happening during the summer. This chart is a little messy, but in some years, the summer losses are even exceeding the winter losses. And while there are a number of factors that are impacting bee populations, climate change, habitat loss, parasites, pathogens, other pesticides, We are particularly concerned about the neonics for three big reasons. The first is that neonics are super duper toxic to bees and other insects. They're among the most potent insecticides ever created, several thousand-fold more than DDT, depending on what chemical you're looking at. And to give you a sense of scale, just the coating on one neonic-treated seed has enough active ingredient to kill about 250,000 bees. And in fact, by some estimates, neonics have made U.S. agriculture close to 50 times more harmful to insect life. And if you can see, that harmfulness really begins to spike in the mid-2000s, almost exactly at the time that we start to see these sudden mass losses of honeybee colonies. And again, these other factors are important: climate change, habitat loss, parasites, pathogens, but we don't see other factors that map quite as well with those sudden losses of bees, other than the sudden uptick in use of neonics. And I would note too, neonic use begins in the 90s, but it's really their adoption on corn and soybean seeds where we start to see this harmfulness take off. Second problem with neonics is they are very good at contaminating whole ecosystems. And I think a lot of the speakers have touched on this. So they're systemic insecticides, and they're designed to permeate plants. Unlike your grandfather's insecticides where you spray it on a plant, it kills the bug, and ideally it dissipates, although I guess not with DDT. The idea with neonics is they permeate plants and they make the plant itself toxic to insect life. That means they can be applied in interesting ways, and I'm sure I'm not the first person to show a picture of neonic-treated seed, but the idea here is as that seed is growing into a plant, it literally soaks up the pesticides through its roots and becomes toxic, all parts of it. If it's a flowering plant, that includes the nectar and the pollen. The problem is only 2 to 5% on average gets into the target crop. The rest is either released as dust, and that can actually be higher than 1% in a lot of cases. But the vast majority of it stays in the soil where it persists for years and years and years. Other speakers have talked about how mobile these pesticides are. Anytime it rains, anytime there's irrigation, if it's a lawn application, anytime you water your lawn, those neonics are moving through the environment. They are contaminating new soil. If there's plants in that new soil, those plants soak up the pesticides, they become toxic, pollen, nectar. If there's a water supply nearby, that water supply becomes polluted. Third problem with neonics is they are just all over the place. Certainly, by far, the use on corn and soybean seeds and other field crop seeds is the largest use, somewhere in the neighborhood of 80 to 90%, most likely. But they're also all over lawns, gardens, golf courses, you name it. And to give you a sense of scale here, and this slide has a lot of caveats here, this is the use of imidacloprid in 2014, the agricultural use. I know 2014 is not the most recent year, but it's the most recent year we have data for the use on seeds. And again, that's 80 to 90% of the use, so we have to include that. Imidacloprid is just one of 5 neonics approved by EPA for use across the United States, just one of 3 used on neonic-treated seeds. So again, a partial picture. And actually clothianidin and thiamethoxam much more used in agriculture these days. And if you look, some of these white spaces around Chicago, Detroit, Milwaukee, those would be filled in if we were looking at the non-agricultural uses. Again, very extensively used in those non-agricultural spaces as well, lawns and gardens and golf courses. But what this does give you a sense is that these are used on a vast scale, and it's a potentially unprecedented one. So the total use of the top 10 insecticides in 2001 across the country, the land area that they covered, was about 26 million acres. We know that neonics cover at least 150 acres of U.S. agriculture, so that's a 6-fold increase in area. And again, these are some of the most potent insecticides ever made. And to give you a sense of scale here, again, one neonic-treated seed, enough active ingredient to kill 250,000 bees. Multiply that times 30,000 seeds per typical acre of corn. Multiply that by 90 to 100 million acres of corn that use these seed coatings every year. And what's happening is we're using these pesticides in the same areas year after year after year. The vast bulk of that toxic balance remains in the soil and builds up year over year over year because it persists and spreads out every time it rains to get into every nook and cranny of our ecosystem, soil, water, plant life, wildlife. So what is the impact of that on biodiversity? I'm going to go through this quickly because a lot of the other speakers covered this, but neonics are definitely killing bees. They're definitely impacting bees. That includes honeybees. This is actually from a study funded by industry itself that found the use of neonic-treated seeds harmed honeybee colonies as well as wild bees. We also have a good news study that came out just a couple of months ago linking the use of insecticides, in particular neonics and pyrethroids, to the loss of wild bees and wild bee diversity across the United States. The same is true for other pollinators, like butterflies. This is another study that we got within the last 6 months. It was amplified in the New York Times. And what's so important about this study is, unlike a lot of others, it measured the impact of different pesticides, including different insecticides, versus climate change and land use. And what they found is that of all of these different competing factors that we know are stressors for insects, only one factor rose to the top of the list, and that was the use of insecticides and neonics in particular, especially when it came to monarch butterflies, and in particular the use of neonics as seed coatings, which is not surprising. I think other speakers have talked about the impacts to invertebrate life. It's worth noting that these pesticides are not just impacting bees, they're not just impacting butterflies. These are the species which we have better data sets for, right? People like butterflies, people go out and count them. We have good data on butterfly species, on honeybees, on a number of wild bees. We don't have it for a lot of other insect species, but we do know that we're losing insects. We know there's a lot of causes for that, but neonics are sort of a leading preventable cause. And we see that other places in the food web. So a lot of birds eat insects and require insects to live. So we see associations with neonic use in the loss of birds and bird biodiversity. Unsurprisingly, most pronounced in grassland species where have habitats in agricultural areas, and also insect-eating species. I don't know if folks remember this study or know about it. It came out about 5 years ago. It was amplified in the New York Times. We've lost 3 billion birds in the last 50 years, 30% of our bird species in North America. This study didn't look at neonics in particular, but a couple of scientists went into that data set later, and really interesting finding here. So they divided the data set into birds that require insects at some point in their life cycle versus those birds that don't. And if you look, the birds that don't rely on insects are actually doing pretty okay, and in some cases have increased populations in the last 50 years. Unfortunately, the birds that rely on insects are getting hammered. And as it turns out, that's kind of the vast majority of birds. Same thing happens in aquatic ecosystems. So this is an interesting study, came out a few years ago from Japan, where they were studying zooplankton. And I always put this slide up because a lot of people don't know what zooplankton is. I'm sure about half the room here does. But for those who don't, these are zooplankton, essentially water bugs. And those authors, it had a long-term data set going back to the early 80s. And they noticed in the early 90s that the zooplankton population they were studying basically hit a wall. And at the same time, this was in a sustainable, otherwise sustainable fishery, catches of fish species that ate the zooplankton, relied on them as a food source, unsurprisingly also declined pretty sharply at the same time. The study authors later connected it to the introduction of neonics in nearby agriculture. This is actually one of the first places neonics were used in the world. Japan was one of the first places to register neonics. So a really interesting study, and we see similar levels of neonics in water across the country. So this is just a wacky finding. So EPA last year determined that Neonics are likely jeopardizing 200 plus endangered species. For those who know anything about the Endangered Species Act, it is a violation of that act to jeopardize even one. I don't know of any other finding by an agency that comes even close to this. And even though EPA, we won't get into the technicalities here, EPA doesn't have the final say on jeopardy. The fact that EPA came out and essentially admitted that it violated the Endangered Species Act 200 times however many neonic products it approved, it's a really shocking finding and speaks to the broad harmfulness of these pesticides. Let's not think that all of this harmfulness is on invertebrate species and then indirect toxicity to things with spines. Neonics are also directly toxic, and birds are a good example. We know that one neonic-treated corn seed has enough active ingredient to kill a small songbird. But similar to bees, at levels that don't kill birds, neonics can severely impact their survival skills, their ability to migrate, to find food, to navigate, their immune systems, their ability to reproduce. So even if that ingestion of the seed doesn't immediately kill the bird, it can ultimately lead to the death of the bird or the population. This is from a study showing that neonics delayed migration for white-crowned sparrows. It actually caused them to lose weight very rapidly. Some headlines said that it caused the birds to be anorexic. That's not accurate, but it did have some pretty profound impacts that impaired white-crowned sparrow migration. Oh. So it's just worth noting, too, that birds definitely eat neonic-treated seeds. This is a survey from the Minnesota Department of Natural Resources. They looked at 70-plus fields, found that about half of them had seeds that were available to birds at the surface. They observed several hundred spills, and they observed 11 species of birds and 9 mammals eating neonic-treated seeds at these sites. So we know that, at least from this one survey, it's a fairly common occurrence, and I'm sure it's similar across the country. And mammals, again, exposure to mammals, this is where things start to get a little scarier for people. There was a study done in response to observations in the upper Midwest that deer that had been culled, there were strange things going on, pronounced overbites, strange things going on with the organ sizes and genitals. There was the thought that this may be related to the use of neonic pesticides. So a controlled study was done, and where confined deer were fed real-world levels of neonics in their water supply. And what the study found is a lot of these same impacts, right? That higher levels of neonics were associated with deformities of the jaw, reduced jawbone length, decreased organ sizes, that includes the liver, the spleen, and the genitals, decreased thyroxine levels, so hypothyroidism, and increased mortality for fawns. In the adults, lethargy was also observed. And one interesting thing about this study, in the control group, even though it was a much smaller amount, sort of trace amounts, there were neonics in the bodies of the control group. So just sort of speaks to, in our world today, it's very hard to not be exposed to neonics, even when you're trying to control for that. So the refrain from industry after that study came out is, well, you can feed a deer enough Mountain Dew and find impacts, right? But deer aren't drinking Mountain Dew in the wild. They're not being exposed to these levels of neonics. And Minnesota Department of Natural Resources did an interesting survey again, where they asked deer hunters across the state to submit spleen samples to the agency. Imagine 800 deer spleens moving through the mail. But those were analyzed, and they found in 2019 that 61% of the spleens had neonics in them, 29% at levels associated with higher rates of birth defects and death in the Burheim study. They did that same survey 2 years later, and wow, what a difference 2 years makes. Those numbers jumped to virtually all deer. 94% of deer had neonics in their bodies. Didn't matter where they were caught in the state, right? Whether that was an urban area, suburban area, agricultural area, whether that was in the boundary waters next to no other human development or not much. And nearly two-thirds at levels associated with birth defects and death in the Burheim study. Where things get a step scarier is we see sort of a similar pattern in human exposure. So in 2019, CDC published the results of biomonitoring. This is thousands of Americans found that on any given day, neonics were in the body about half the American population. And they know this because people are literally urinating neonics or their metabolites. We see in a more recent study of pregnant women across the country that over 95% of those women, again, a much smaller sample size, but still strong results, over 95% had neonics in their bodies, and that those levels were steadily increasing both in frequency and magnitude from 2017 to 2020, and with the highest rates in Hispanic women. And what is also concerning about that is that we know from population studies, you can't do controlled studies with people, that but for exposed populations, inadvertently exposed, controlling for other factors, we see higher rates of birth defects of the heart and brain, higher rates of autism-like symptoms, cognitive impairments, and then in adult exposures, a host of both neurological and sort of hormonal sexual impacts, very similar to what we see in the Berheim study. I'm going to try to quickly highlight this paper. It sort of points out why we may be seeing this. Again, as James mentioned, neonics mimic nicotine, right? The name neonicotinoid is new nicotine-like substance. The receptors that neonics target are very well distributed through insect bodies, but we have them in very sensitive areas of our brain and central nervous system. Thanks to evolution, there's a lot of the same machinery there at the back end. And this is a study that came out recently looking at the tests that industry submitted to EPA to prove the safety of neonic products. But what we see here, even though there are some behavioral tests and we see some impacts from neonics on rodent behavior, as our staff scientist, Dr. Sass, likes to say, you can't really ask a rat how it does on its, or how it did on its second grade math test. But one of the really scary things about this, these tests, these studies, is that we're seeing for all of the neonic chemicals impacts to the size of rodent brains, right? Observable reductions in critical areas of the brain. And these, even though you can't show causality, there's a correlation to ADHD in similar areas of the brain that are reduced in folks that have that. But I think it is pretty self-apparent that when you have a chemical that is reducing the size, the actual physical size of critical areas of brain, that's a major concern. I'm going to come up here again and I'll talk about why we think EPA is not paying enough attention to those results. And very quickly, because I'm already out of time, just how do people become exposed to neonics? We know neonics are in drinking water. Most systems don't test for neonics, but where they do test, we've seen them in Long Island, we've seen them in Iowa. I'm sure they're in more places, but we just don't know because most drinking water systems don't test for them. What we do know is conventional chlorination does not remove neonics from contaminated water. And we know that most water systems don't use the sort of advanced filtration that would remove neonics. Also worth noting that neonics are in produce, they're in baby food, they're in applesauce. This is just one study, but there are others. And they're particularly problematic from that standpoint because they're systemic, right? So if you have an apple treated with neonics, good luck washing them off, right? They're in the apple. So particularly difficult to avoid those exposures. And it's also worth noting that those exposures are unequal. You can avoid a lot of neonic exposure by buying organic produce because neonics are not allowed in organic produce. However, because neonics migrate, there are some tests that find neonics even in organic produce, but obviously at lower levels. You can filter your own water. That's obviously not available to everyone. We also see disparities in terms of who's exposed to neonics. And it's worth noting, as some other speakers pointed out, we're already producing less of certain crops because we have pollinator limitations because of a loss of pollinators. If these trends continue, a lot of those foods are going to become scarcer or more expensive, and that's going to hit folks least able to afford it.
Great. Thank you, Dan. And so if all three speakers could make their way back up to the stage, and we can have a little time here for Q&A, I believe. AJ, do we have any questions online from this session, or should we start in the room?
Don't currently have any online questions.
Great. All right, questions for the room.
Thank you so much. I'm just curious, when you talk about advanced filtration, being able to remove that, what does that look like for folks? Is it like carbon?
Yeah, that's granular. Can folks hear me?
Yep.
Yeah, granular active carbon filtration.
Good. So if we're treating for PFAS, we're also treating for neonicotinoids.
Yeah.
Yay.
Yeah, and I think because of PFAS, you're going to see those systems in place a lot more places. But yeah.
So, James, should we stop eating honey?
It's good. I mean, I think those levels, before 25 minutes ago, I would have said those levels are well below what we should worry about as humans. And is this coming through? Can you hear? No. Hello. Is that
That's
okay?
better.
Sorry, closer. Yeah, I would have said well below levels for, for humans. So I generally don't consider, at least, yeah, those levels are quite low, on the lower end of what is necessarily directly toxic to bees, hopefully below what has human impacts, but I've just updated my prior on that a little bit. Yeah, so I mean, I think it's really hard to know. We probably should, the overall amount you're going to get through honey consumption, probably lower than like general food consumption, but something to be concerned about, for sure.
Other questions? Oh, AJ's got one on the back of the room. Go ahead.
So this is for all the speakers. Any studies that link the decline of common nighthawks specifically to neonics?
Ooh.
Not that I know of.
Not that I know of.
Thanks, AJ. Go ahead.
Okay, this is a question for Gavin. On one of your slides, you had a flow chart of where the neonics are and where they go, and I saw wastewater treatment plants on there. Is that a commonly known source of neo? I mean, obviously that's not the source, but source to the rivers.
That's it.
I know we see a lot of different contaminants coming out of wastewater treatment, like PFAS and pharmaceuticals and such. Are neonics on that list now?
It's a good question. I know off the top of my head, I would say that they should be if they're not, but I don't think it's a main source of what's being added. I think it's just a way for us to understand the levels that are coming out, if that helps.
Yeah, and I can speak to that a little bit too. So neonics are very popular in pet products as well. So like dog shampoos and stuff, any sort of flea control. So, you know, we used to think that was maybe sort of an insignificant addition of neonics to the environment, but I think what we're beginning to see from wastewater treatment plants might suggest otherwise.
Great. Good question. AJ.
Online. The first is, are there any studies showing links between levels of neonics in surface water and the impacts to terrestrial invertebrates?
Yeah, I mean, there's definitely good studies showing overall neonic levels and yeah, declines in terrestrial invertebrates. I don't know, and this just might be that I don't know them. I don't know of one specifically looking at sort of mapping surface water contamination directly to, per se, to terrestrial insect declines. But we also, one thing I want to say is we know that there are routes of exposure from surface water to, so for example, like if you have a pollinator meadow next to a corn field, right? I mean, one of the ways we've thought is, oh, great, well, the neonic is in the corn, but pollinators aren't visiting corn, so what's their actual exposure level? Well, now something we know very well is through groundwater, those neonics move into adjacent wildflower plantings, so you can totally find neonics in the nectar and pollen of adjacent plants to which they were not applied, which is just one example of how that surface water is likely moving to adjacent habitats. So I think it's likely that that link exists.
Yeah, and I don't know about terrestrial invertebrates, but there is a study from Europe in 2014, Hallman et al., that linked surface water concentrations of neonics to the loss of birds. And I forget which species of bird it was, but yeah.
Great. The second question is there anything positive we can say about how neonics are being addressed in the United States?
I'm going to talk a little bit about that in the next panel, but yeah, there is some progress at the state level.
Yeah, and I just want to highlight that I think some of the work we heard about earlier is really like showing evidence for places where maybe we don't actually need as much and there's not as much benefit to neonic use as we maybe once thought. I think that's been really critical for driving some of the state level action, like in New York. So I think the generation of those data, I think, are really promising also for actually making the case for at least reducing the use of neonics.
I'd like to ask the NRCS man, who I believe you're the only person, do I have you wrong? You, the one you're looking around.
NRDC.
Thank you. I think you're the only person who mentioned that by eating an apple off a tree, you're likely eating neonics. I have the same question about potatoes. Every potato I eat is probably a potato grown in Marquette County, and it's probably from some beautifully colored seed or something. Am I eating neonics?
So I think it's hard to say, right? So I just showed one study from Friends of the Earth, which had some pretty high levels. What we've seen in conventional produce is that they're not necessarily in all conventional produce. They're in a lot, and they're in certain products. I haven't seen testing for potatoes. Apples, I know, tend to have higher levels of neonics. So the answer is I'm not sure. But I think we do need to do more testing for these pesticides in produce.
Great. Okay, and with that, I think that kind of concludes our Q&A period for this session. So now we are going to roll into a slightly longer break. So we will break until 2:45. So we've got about 20 minutes in this one. So we have some more refreshments out there up for grabs, so please have a chance to do that, intermingle. And then we're going to move into our last session of the day, which is going to hit on some of the things that I know have also come up in a few online questions that we haven't quite addressed yet, is what is EPA's role in this? They're going to be our first speaker. Then I'll come back and talk a little bit about action at the state and things we've been doing so far and where we're headed with a few. And then Dan's going to wrap us up for the day to talk about what other things have been happening across the country and the globe in this realm. So thanks for joining us, and we'll see you back in 20 minutes.